I'm not muted my mic. It's in unmute mode. Sorry, why it's happened. Our neurotransmitters are responsible for passing signals from one cell to another cell. This signal exchange between cell to cell is responsible for the cell multiplication and cell activation. So only we are going to study about the neurotransmitter. Before the neurotransmitter, we want to learn some basics. That basics concepts are I am going to explain now. These are all the topics today we are going to cover. First is how they communicate between each other and about the cancer cells. Okay. First, major cells are produced in the blood. They are activated in the blood circulation. So first we need to know about how they are produced in the human body system. So that concept, the production of blood cell, is the basic concept, is called as the hematopoiesis, the production of blood cells. Okay. Blood cells are produced from, or usually from, the stem cells. Stem cells, we all know stem cells are the major thing which helps to improve and produce and multiply all the cells in this stem cells. Okay. In this hematopoiesis process, the blood cells are produced from the stem cells. Okay. After the stem cell production, a small amount of stem cells are stored in human body itself, and after some particular time, that are called as the progenitor cells. Progenitor cells means that are the initial cells which helps to produce all kind of cells. It's one small process, but it having more value. In this hematopoiesis cell, first major cells produced from the first process, that is called as the T cell B cell production, because that are most important cells in our immune system. Apart from the T cell B cell, progenitor cells are divided into the different blood cells, that is the basophils, eosinophils, neutrophils, and erythrocytes, platelets, macrophages. All these cells alone involved in the cell communication immune system, because in our human body, various parts having various tissues. All cells are communicated with only the blood cells. Except neuron cells, other cells are communicated with the blood cells. It's a basic concept. Then another important thing we are going to discuss about in this hematopoiesis, that is, first slide is a basic introduction about hematopoiesis. Next step is what are the called in the involved in the hematopoiesis process. First, the hematopoietic stem cell is originated in the embryonic stage. Then the lymphoid progenitor is there. Another one is myeloid progenitor. These lymphoid progenitors are act signals of TNF alpha, TGF beta, interleukin 2, 7, 12, and FLTs. These are all the signaling compounds of lymphoid progenitor cells. This lymphoid progenitor cells get the signal in their external source and they are alerted. Okay, something happen in our human body, that are produced the lymphoid cells, lymphocytes. These lymphocytes are responsible for the our immune system response and cell communication. The question here is, what are the signaling compounds which are responsible for the lymphoid progenitor cell? They are not direct question. Give the paragraph, give the paragraphic explanation. In that they mention this are the this chemical compounds name, that is FLT 3 ligand, STS 1 TNF alpha TGF beta, that kind of words are they mention in the particular line. Okay, and the next is a myeloid progenitor cell. In this myeloid progenitor cell we have 4 different branches. Myeloid cells are produce thrombocytes, blood cells, eosinophils, mast cells and erythrocytes. The different cells are produced by different signals. Okay, thrombocytes are produced based on the SCF, GM, CSF, IL-3, interleukin 3. Very important thing what we want to understand in this myeloid cells means all kind of erythrocytes, erythrophils, mast cell, erythrocytes, monocytes, basophils, neutrophils and thrombocytes all are produced based on the interleukin 3 signaling. Interleukin 3, what is the role of interleukin 3 in this myeloid cells? Because these myeloid cells are observed and understand the signals from this, okay, understand receiving the signal from interleukin 3 and produced the blood cells and activate the blood cells. It's a very important thing in the hematopoiesis. Hematopoiesis having the different 2 progenitor cells, that is lymphoid and myeloid cells. The both are activated by the different signals. That signals are produce the blood cells. Okay, next is the NF kappa B activation. Here all of the cells are activated and generated based on the that activity happens because of the NF kappa B process, NF kappa B activity. This NF activity is denoted and explained based on the our hematopoietic cells, that is common myeloid cells and progenitor cells. Okay, the myeloid cells are activated by the P65 and the CREL gene. 2 genes are helps to activating the upcoming. The previous slide we are explained only the signaling compounds, and in this slide we can see I'm just explain what are the compounds, signaling compounds, what are all the cells are hematopoiesis, and here I'm going to explain what are all the proteins involved in the hematopoiesis stem cell in the hematopoiesis process. Okay, here they are asked the question related to the P50, P65, that kind of aspect. So just observe the concept. The major role playing proteins genes are P65 and CREL. Okay, in the osteoclast they are having the T and P52 compounds. Macrophages are based on produce the response based on the NF kappa B, granular response for NF kappa B. That dendritic cells are produced by the myeloid progenitor cell. That dendritic cells are responsible, they are activated by activating some genes, erythrocytes. The same process happen in the lymphoid progenitor cell. In the lymphoid progenitor cell we have 2 options. One is pro B cell, pro T cell. Okay, pro B cell pathway is all about the NF kappa B. only way the pro B cells are activated the pathway NF kappa B, that is not activating any other pathway. But this T pro cells are T pro sorry pre T cells, pre T cells are activating the CD4 and CD8 cells. That CD4 and CD8 cells are activating the CREL and CRLB, that compounds. Here focus, the main underlying concept is what are all the cells are activating the NF kappa B pathway. So I'm going to end up with the NF kappa B concept, because all of the pathway, all interlinked, hematopoiesis pathway is interlinked with this NF kappa B concept. So only I am explaining this. Next is the steps. Again, just I'm reminded this. What are the steps involved in the signaling? First is receiving the signal at the cell membrane. That relay happens in the molecule, then activate the effector, then gene transcriptions. We all know about these steps. Once again reminded this, because these are all the process are involved in the upcoming cancer cell line pathways. So only I am explaining this. Please observe the concept. Signaling molecules, that hormones, okay, these hormones are observed at the cell surface receptor, that after the relay happened, the transduction process happened, that gene transcribed based on the received signal. Next concept. Okay, now enter into the cell adhesion molecule. Once how they receiving this signal from the external source. This external source signaling is received on by the cell adhesion molecules. These cell adhesion molecules are the adhesion molecules are present in the cell surface. These cell surface proteins are transmitted through the membrane. This membrane having three different domains. One is extracellular domain, transmembrane domain, and intracellular domain. These three are major part of the cell adhesion molecule. Okay, this intracellular molecule is with the cytoskeletons, and the extracellular compounds are having the intracellular domains with the cytoskeletons, and transmembrane domain with the adhesion molecule. The adhesion molecules playing a role at the position of the molecule and transmembrane molecule. This transmembrane is explained with the proteins. All the proteins are interlinked in this intracellular membrane, because the external surface membrane having external surface of the cell membrane having the that may be activated by the particular signals of other cells. This other cell signals helps to understand what happened on the transmembrane. The changes, it makes some changes in the transmembrane. After getting the membrane, this extracellular that intracellular domain and extracellular membrane will open or close for the response. The another next concept now I am going to discuss about the in the cell adhesion molecule is role. It involved in the lymphocytes formation. So this cell adhesion molecules are activating the lymphoid cells. Then another thing is involvement of immune response and immune regulation. Then another is involvement in the inflammation. Once our lymphocytes are activated during signaling, then automatically our immune system will get alert, so it change its activity, the change produce immunity in our human cells. And then major concept, this cell adhesion helps to regulate the indirectly it does regulate the cell adhesion process. Okay, what are all the compounds which major role playing in the cell adhesion molecule, cell adhesion molecule in the cell process? We are going to discuss 4 different molecules. These are all the well studied and proven molecules. These molecules are repeatedly asked in the CSIR questions. This table is very important. Please understand, I am telling this again and again, means the questions are indirectly focusing on based on this table explanation. The Catrins, this having 3 different membranes, E, P, N. This compound, this cell adhesion molecule having the homophilic and hydrophilic interaction. It's regulating the hypermethylated in leukemia. Experimentally they are proven the anti-B-catrin in the memory cells. They are already available in the our memory cells. Another thing is Ig, the immunoglobulin. This immunoglobulin is a superfamily cell adhesion molecule which having different members. These members are having again 2 type of address, homophilic and hydrophilic. Important thing is regulated by RUNX1 protein. It's a major coated thing. This Ig superfamily cell adhesion molecule activated by the RUNX1. Then another thing is mucin like cell adhesion molecule. This is one thing, hydrophilic compound. Please understand this is the hydrophilic compound. This hydrophilic compounds having different p-selectin molecule. This p-selectin molecule overexpressed in the multiple myeloma. Another thing is integrin. Integrin is present at the cell surface and the cell outer layer. It's hydrophilic. It activated via CDF1 alpha and CXCR4. Please understand the table to next slide, because here we face many internet connection. So I am firstly move to next slide. The major concept from this slide is how they interact, how they activate. It's a major thing. Okay, gap junction. What is gap junctions? These gap junctions are expressed in the cell membrane. Why they are present at the cell membrane? These gap junctions are of it's a G coupled protein. Receptors are made up of the connexin subunits. This channels helps to transfer the signals from extracellular membrane to intracellular membrane. Action of the particular gap junction proteins, we can realize the feedback, negative feedback activity, that kind of process are happen because of only gap junctions. Okay, what are the parts and membrane systems are in the gap junctions? This gap junctions having the protein which is having N terminal end and C terminal end. This having the cytosolic loop, connexin domain. Connexin domain. What is connexin domain? This connexin domains helps to observe what are the signals are we need or we don't need. If it is needed for the cell process, it will absorb, otherwise it's not allow into the human cell membrane or cell system. It's a major concept. Okay, how it's look like. Okay, this is the cell, the gap junctions formed between two cells that are look like this. Here the another representation, they are explained with the image where the gap junctions are arranged. If it is in closed mode, all the proteins are tightly packed. If it is in open mode, if it is allow the signals from one cell to another cell, it's in open mode. The size of gap between the cell to cell is below 4 nanometer. That hydrophilic channels, must be the hydrophilic channel, because the cell surface is made up of the lipids. This gap junction is formed like the hydrophilic channels. It's a major things, please understand. Okay, now after that are the normal cell communication. Here now enter into the neurotransmission. Why it's happen in the neurotransmission? Because this neurotransmissions alone are activating all the cells. This happens because based on the 7 steps. The action potential is reaches the presynaptic terminal. Presynaptic terminal means it's one that cells receiving the cells initial part is allotted by the activity. We want to produce the signal for other cell. That is the signaling. Okay, just allotting. Then another is second step is a depolarization of the synaptic terminal, opens ion channel. This allowing the calcium ions into the cell. After the getting alert that okay, we want to produce the signal, so our cells are allowing the calcium channels to enter the calcium ions in cell. Calcium's plus ions triggers release of neurotransmitters from the vesicle. Vesicle is the that presynaptic terminal. They are the vesicle. They want to that they are having the vesicle. So the calcium is present in the presynaptic terminal is alert our vesicles. We want to transmit the signal. The fourth step is transmitter binds to the receptor binding site on the postsynaptic membrane. Postsynaptic membrane is the opposite one. Presynaptic is the signal transmitter. Prosynaptic is the signal receiver. The neurotransmitter binds to the receptor at the binding site of the postsynaptic membrane. This postsynaptic membrane are ready for opening and closing of the channels cause the postsynaptic membrane potential. That process is opening and closing. If it is in the intracellular, the outer membrane protein is the receptor is closed. If it is closed in the intracellular membrane, the extracellular membrane is ready to receive the signals from presynaptic. This after this step, action potential propagate through the next cell. Okay, now the signals are transmitted through the prosynaptic channel receptors. Now our prosynaptic cell received all the signals from the presynaptic, and the neurotransmitters are inactivated. After the cell receiving the information passed to the prosynaptic cells, our neurotransmitters is inactivated or transported back into the presynaptic terminal. It's 7 steps. Based on this story, they are asked many questions. Okay, for example, they are asked if the calcium ions are higher quantity in the presynaptic cell. What is the what is the potential action potential of the prosynaptic cell? If the calcium cells are higher in presynaptic, they are activating the vesicle in a higher manner, they are producing a more number of signals, so the action potential of the prosynaptic cell also high. It's a correlated information to the neurotransmitters. Okay, now enter into the cancer cells. What is the cancer cells? It's a proto-oncogene that called as the that activated by pathway. Oncogenes are the RAS pathway is a major role playing in the cancer oncogene activity. If normal RAS pathway is activated in a normal mode, if it is mutated, that is act like a cancer cell. RAS activated cell, it's in this oncogenes are always in activated mode. It's stimulating in many signals, and also it's proliferated in the control manner. If it is an out of control, if it's having pathway disturbance, that will that is produced in the larger number. Okay, what is the progenitor? In the previous blood cell hematopoiesis itself, these progenitor cells are responsible to produce many cells. That may be the stem cell, specialized cell, any cell. It can have the capability to produce. It may be produced as a neuronal system. This the progenitor cells are specialized cell, that neurons or the astrocytes. These are all the things happen in the progenitor cells, so it's very valuable in the immune response. What is the genetical changes happen in the cancer cell and normal cells? The chromosomal abnormalization. The cell abnormalization is now abnormality is called as the cancer cell, because normal cells are following in the flow, but cancer cells are not in the control. So we need to understand whether the chromosomal abnormality is happen due to the genetical variation in cell because of the cancer causing agent. The balanced chromosome rearrangement, how they happen in the cell. Okay, in normal cell, the DNA is producing some protein, and that proteins are in the controlled manner. That control, because of the proteins that signaling pathways are activated, that again helps to our chromosomal and DNA replication. Okay, if the person is affected by the cancer, means some pathways are altered due to the cancer causing agent. This alteration make changes in genetic replication. That there are missing of many genes in the, or mutation of many gene in the, or during the replication process, that is make the imbalance in the chromosomal activity, chromosomal count. This imbalance means in normal gene, additionally we may get, or we may lose any chromosomal part. That is called as chromosomal imbalance. This chromosomal imbalance happens due to the cancer causing agent activity in particular pathway. This image is going to explain how they are multiply and survival of particular cancer cell. Acute transformation by the retrovirus. This retrovirus is the major concept involved in the cancer experiments. because it process other viruses are not studied well. In acutely transforming retrovirus, once enter into the human cell, that is integrated in infect the human cell, then only the viral particle alone enter into that integration of viral happens in the our human cell system. After the integration, this neoplastic transformation happen in the cell. That neoplastic is make the cell into oncocell, that is become a cancer cell. These are all the steps involved in the acute transforming. In this process, they ask many questions relevant to the steps. First is a normal cell, then infection of host, after the infection in the neoplastic formation. Neoplastic formation related questions are most repeatedly asked in the examinations. The CSIR examination, they are given like a story. Then that is acutely happened process, slowly transforming retrovirus, how they are slowly transformed into process. In this slowly releasing process, the reverse transcriptase is a major thing happen in the involved in the cancer cell production, neoplastic formation. Okay, apart from this, it's a major question they are repeatedly asked in the CSIR examination. Please listen, the tumor suppressor genes. It's a most repeatedly asked question in CSIR examination based on these four things. They are asked about they are mentioned a big paragraph, but that concept is one line concept. They are mentioned based on the basic things alone, but they are given as a paragraph, big paragraph. Okay, what happened in the tumor suppressor gene? If the gene RB is having a 13 chromosomes, it's present in nucleus, it affecting the cell cycle. A cell cycle means it affect the phase growth, cell growth happen at any step. This, based on this information, they ask the question. And P53, they are the hero in this tumor suppressor process, and all the questions without this P53 word, you not seen any question. At least you may seen it P53 word in or 1 or 5 to 6 words, 6 times. They mentioned repeatedly about the P53. You can see the questions with the P53 rapidly. So a study about this process. This P53 helps to DNA repair and apoptosis. Then BCL2, that responsible to it's a gene. It's producing BCL protein, which helps, it's present at the mitochondria, helps to apoptosis. Okay, here the question what they asked means we want to change the cell cycle. For this, altering the BCL2, that is producing activating the nucleus membrane and nucleus cells. If it is a question, you must confused about the question. Okay, we studied about the BCL2, how it activating the nucleus. It's a question first rise in our brain. We want to realize what happened in the question, what they are asked in the question. They are confusing us to get a clear answer. From the all of the questions are easy when you are understand the questions are easy when you understand the answers, because most of the CSIR questions have the answers in the question itself. That questions are indirectly expose the answers. So please understand, this is a major topic, tumor suppressor gene, that questions are basically asked from basic concept. This gene location and function. I will send the PPT, please refer this. Okay, in a normal P53 pathway, the apoptosis happen. Another thing, cell cycle, if it is in a normal apoptosis happen, so cell is automatically produced and get death after particular period. That is a normal process. If it is if the 53 mutated in a human cell, or there was no apoptosis though, so the cells are uncontrollably grown in the human. It's an viral based based oncogenes. In this till day we are studied about the human papillomavirus, the Epstein-Barr virus, another is hepatitis B virus of DNA, that are other DNA virus. They are studied for other than some RNA viruses too, that is human T virus and hepatitis B virus. These are all virals can cause the cancer in the human cell. That are all the reports are submitted by the various institutions. Then what is metastasis? Metastasis is a major dangerous thing happen in the cancer patients, because once they are affected in the one particular place, that cells need of more food, because there is the large number of people in particular one place, right? There is a large number of cells in the one particular place, so they are not get enough blood, enough nutrition, so they need nutrition. So that cells are attracting and make a connection to another part, another neurons, another cells, another bones, to somewhere they need make a connection with some other places to get the nutritions. Automatically it will connect the make a neurons, make the cells, and make the nerves or cells. That cells are help them to move from one place to another place. They are moved for the getting food for the survival. In a primary tumors, from the primary tumor site, that is the first place which cancer caused. From that they are automatically move through the bloodstream, that and enter into any part. It can enter into any part by the cell epithelial cells, because you can see here, this is the cell present primary site, the damaging the epithelials, the epithelial cells in the nerve system, right? Now our blood system having the so epithelial cells at their external surface. It break down this cells and enter into the bloodstream. After entering into the bloodstream, it's move towards various parts and enter into these again damaging the epithelial cells and enter into the another part. Okay, this is the apoptosis pathway. Here I wish to mention some things, but this is not clear, I think not clear to your display. Why I don't know it's happened. The many of the images are not clear, so please observe my words. First thing happen in the apoptosis process at the cell surface receptor. The cell surface receptors are receiving some signals, ligands from the cancer causing agents. These signals are affecting the caspase-8 pathway. This caspase-8 path helps that if the signals, the chemical compounds affect the cas8 pathway, automatically cas3 pathway will get affected. The misleading of this caspase-3 pathway in the apoptosis. This if their cell is not undergone apoptosis, that creating cancer in the human cell. It's a one pathway. Another pathway is the mitochondrial cells are for the caspase 9, 3, 7, 12. These are all the some signals, extra signals are produced in the human cells. That cells are responsible for apoptosis. So apoptosis pathway having different caspase. Without this caspase word, you are not seen any question. All the questions papers of CSIR having this caspase word, because it's a very very important concept of cancer and metastasis, tumor suppressor gene related questions. Please go through some materials. I'll send it in your Google Classroom and mail ID, because many of the questions rapidly they are asking the questions related to this tumor suppressor gene space questions. All questions are like the experimental questions. Experimental questions they are asking the, for example, one cancer patient is treated with the apoptosis inducing compound. How will you estimate whether it is induced apoptosis or not? We are not directly optimize how the quantity happen, how the quantity of cells are undergone apoptosis. For that we are need to understand how the proteins are produced in the particular cell. If the apoptosis is happen in the particular cell, happen in a human cell due to the injected molecule, injected apoptosis inducing molecule, the caspase 3 and 7 is activate automatically. So if you are estimating the caspase molecule, you can understand, okay, apoptosis has happened in the cell. It's the basic thing. If the quantity of measuring of the quantity of caspase 9 is one thing which is responsible for understanding apoptosis process. Another thing is the therapeutic values, interventions. Many of the therapeutic interventions are available in the market for cancer treatment. Thank you so much. Next, tomorrow we will discuss about the immune response related question. Tomorrow we will discuss the immune response related immune and immunological related concepts. In this chapter today we discussed neurotransmitters and cancer cells. Notes related to this topic I'll send to your mail IDs and Google Classroom along with your yesterday material. Thank you so much. I think we met many problems in the net connection. Thank you for your patience. Thank you so much. Now I am going to end this. Queries, please post it in YouTube channel. Okay, no problem. Menaka